Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Semiconservative DNA replication, prokaryotes versus eukaryotes explained. Table of contents. Introduction to semiconservative DNA replication. Enzymes involved in DNA replication and their functions. DNA replication in prokaryotes. Proofreading of replicated DNA. Evidence for semiconservative replication of DNA in prokaryotes. DNA replication in eukaryotes. Proofreading. Evidence for semiconservative replication of DNA in eukaryotes. Difference between prokaryotic and eukaryotic replication. Introduction to semiconservative DNA replication. In the semiconservative model of DNA replication, two identical copies of the original DNA molecule are produced, each containing one original parental strand and one newly synthesized strand. Watson and Crick's double helix model of DNA includes an integrated template system that enables self-replication or autocatalysis. Due to specific base pairing, adenine with thymine, and guanine with cytosine, the sequence of bases on one strand determines the sequence on the complementary strand. Each strand of the DNA double helix serves as a template for synthesizing its complementary strand. Watson and Crick proposed that replication begins with the disruption of hydrogen bonds between base pairs, followed by the rotation and separation of the two polynucleotide strands. Each purine and pyrimidine base on the parent strand attracts its complementary free nucleotide in the cell, holding it in place through specific hydrogen bonding. Once held in place, the free nucleotides are joined together by phosphodiester bonds that link adjacent deoxyribose residues, creating a new polynucleotide strand with a specific base sequence. The outcome of replication is two double-stranded DNA molecules with base sequences identical to the original DNA. One daughter DNA molecule includes the original left strand, while the other includes the original right strand. Because each daughter molecule retains half of the original DNA, this pattern of replication is termed semiconservative. DNA replication occurs in both prokaryotes and eukaryotes, and while the fundamental mechanism is similar, the process is more complex in eukaryotes. Besides the semiconservative model, two other possible models of DNA replication have been proposed. In the conservative model, both newly synthesized strands form the new DNA molecule, while the two original strands remain together, fully conserved. In the dispersive model, the parent DNA double helix is broken into fragments during replication, and the new DNA molecules consist of interspersed segments of parental and newly synthesized DNA. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Enzymes involved in DNA replication and their functions. DNA helicase unwinds the DNA helix by breaking hydrogen bonds between bases. Topoisomerase alleviates tension in the DNA strand by cutting and resealing it during unwinding. Single-strand binding proteins prevent the single-stranded DNA from reannealing after being separated. Primase synthesizes short RNA primers to initiate DNA synthesis. DNA polymerase I removes RNA primers and fills in the gaps with DNA. DNA polymerase II mainly functions in DNA repair mechanisms. DNA polymerase III, primary enzyme for adding nucleotides during replication. Sliding clamp, stabilizes DNA polymerase on the DNA strand for efficient replication. DNA ligase, joins Okazaki fragments on the lagging strand to form a complete DNA molecule. DNA replication in prokaryotes. DNA replication in prokaryotes occurs in three main stages. 1. Initiation. 2nd, elongation. 3rd, termination. First initiation of DNA replication in prokaryotes. Replication starts at a specific location on the chromosome known as the origin of replication. In Escherichia, coli, and most other prokaryotes, this origin is referred to as Ori-C. Ori-C is approximately 245 base pairs long and contains multiple AT-rich sequences, which are easier to unwind due to having only 2 hydrogen bonds. This region includes repeated sequences, specifically 13 base pair repeats and 9 base pair repeats. Initially, around 30 DNA proteins bind to the 9 base pair repeats, causing the DNA to bend and initiate the unwinding of the helix at the adjacent 13 base pair repeats. DNA-C proteins then help load DNA helicase, DNA-B, onto the origin. DNA helicase unwinds the DNA by breaking the hydrogen bonds between base pairs, a process that requires ATP hydrolysis. As the DNA unwinds, replication forks, Y-shaped structures, are formed. 2 replication forks are established and proceed bidirectionally. The activity of DNA helicase generates topological stress, causing the formation of supercoiled DNA. Topoisomerase resolves this stress by temporarily cutting the DNA helix and resealing it, preventing overwinding ahead of the replication fork. Single-strand binding proteins bind to the separated strands to stabilize them and prevent reannealing. DNA polymerase cannot initiate synthesis independently. It can only extend a chain in the 5' to 3' direction and needs a free 3' hydroxyl group for nucleotide addition. The enzyme RNA primase synthesizes a short RNA primer, 5 to 10 nucleotides, complementary to the DNA strand to provide the required 3' hydroxyl group. Second elongation of DNA replication in prokaryotes. DNA polymerase III is loaded onto the DNA, initiating synthesis. The 2 DNA strands have opposite orientations. One runs 5' to 3', and the other 3' to 5'. The strand complementary to the 3' to 5' parental strand is synthesized continuously toward the replication fork and is called the leading strand. It requires only 1 primer. The other strand, complementary to the 5' to 3' template, is synthesized in short stretches known as Okazaki fragments and is referred to as the lagging strand. This strand requires multiple primers. A sliding clamp protein ensures that DNA polymerase remains attached to the DNA during synthesis. DNA polymerase I removes RNA primers using its exonuclease activity and replaces them with DNA nucleotides. DNA ligase then seals the remaining gaps between fragments by forming phosphodiester bonds. The 2 replication forks continue to move in opposite directions around the circular chromosome. Third, termination of DNA replication in prokaryotes. Termination occurs when the 2 replication forks meet, resulting in 2 complete double-stranded DNA molecules. A termination protein binds to specific tier sequences to block one of the replication forks and halt further progression of DNA helicase. The second fork stops when it encounters the first. During replication, the circular bacterial chromosome can become entangled or linked due to crossover events. Topoisomerase 4 separates these interlinked DNA circles, a process called decatenation. Finally, the two DNA copies are distributed into two daughter cells during cell division. Proofreading of a replicated DNA. Proofreading of replicated DNA involves scanning the ends, termini, of newly synthesized DNA chains to detect and correct errors during the replication process. This proofreading mechanism is carried out by the 3' to 5' exonuclease activity of DNA polymerase. It functions by removing incorrectly paired nucleotides at the 3' end of the growing DNA strand, allowing the 5' to 3' polymerase activity to then add the correct nucleotide. In bacteria, all 3 DNA polymerases, Pol Y, Pol II, and Pol III, are capable of proofreading through their 3' to 5' exonuclease activity. When DNA polymerase detects a mismatched base pair, it reverses direction by one base pair, excises the incorrect nucleotide, and then resumes forward synthesis after inserting the correct base. The efficiency of proofreading directly influences the mutation rate during DNA replication. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Evidence for semiconservative replication of DNA in prokaryotes. The semiconservative nature of DNA replication in prokaryotes was experimentally confirmed in 1958 by Matthew Meselson and Franklin W. Stahl. They used isotopically labeled DNA and an isopycnic density gradient centrifugation technique to demonstrate this. Escherichia coli was grown for several generations in a medium containing ammonium chloride with 15 nitrogen, a heavy isotope of nitrogen. When DNA was isolated from these cells and centrifuged in a cesium chloride salt density gradient, it separated at the point where its density equaled that of the solution. The DNA from 15 N-grown cells was denser than DNA from cells grown in normal 14N medium. The 15 N-labeled E. coli cells were then transferred to a 14N medium and allowed to divide. Cell division was tracked using microscopic counts and colony assays. DNA was extracted at various time intervals and compared to pure 14N DNA and pure 15N DNA. After one round of replication, the extracted DNA showed an intermediate density. This intermediate result ruled out conservative replication, which would have resulted in two distinct bands, one for heavy DNA, 15 nitrogen, and one for light DNA, 14 nitrogen, with no intermediate band. However, both semiconservative and dispersive replication could account for the intermediate density at this stage. In semiconservative replication, each DNA molecule would consist of one 15 nitrogen strand and one 14 nitrogen strand, producing intermediate density. In dispersive replication, each DNA strand would contain segments of both 15 nitrogen and 14 nitrogen, also resulting in intermediate density. After 2 rounds of replication, the DNA separated into 2 distinct bands, one at intermediate density and one at the lighter 14N density. This observation did not support dispersive replication, which would have resulted in a single, slightly lighter band due to uniform dilution of 15 nitrogen in all DNA strands. The presence of 2 bands, one intermediate and one light, confirmed semiconservative replication, where half the DNA molecules had one old, 15 nitrogen, and one new, 14 nitrogen strand, and the other half had two new, 14 nitrogen strands. DNA replication in eukaryotes. DNA replication in eukaryotes takes place in three main stages. 1. Initiation. 2nd, elongation. 3rd, termination. First, initiation of DNA replication in eukaryotes. Eukaryotic chromosomes have multiple origins of replication. Humans may have up to 100,000 origins. At each origin, a pre-replication complex is formed during the G1 phase of the cell cycle. The pre-replication complex assembly includes 6 origin recognition complex proteins, CDC6, cell division control protein 6, CDT1, chromatin licensing and DNA replication factor, 6 mini chromosome maintenance, MCM2 to 7 proteins. Once the pre-replication complex is in place, 2 kinases activate it: cyclin-dependent kinase 2, DBF4-dependent kinase. These kinases recruit CDC45, which in turn brings in other DNA replication proteins. CDC45, MCM2-7, and the GINS complex, Goichi-Nissan, combine to form the CMG helicase, which becomes active in the S phase. CMG helicase unwinds the origin of replication, forming replication forks. Each origin of replication forms 2 replication forks, which progress bidirectionally. The action of helicase creates supercoiled DNA, inducing topological stress. Topoisomerase and replication factor A, RFA, relieve this stress by altering DNA topology. Single-strand binding proteins stabilize unwound DNA and prevent it from reannealing. Primase, tightly associated with DNA polymerase alpha, synthesizes short RNA primers to begin DNA synthesis. Second elongation of DNA replication in eukaryotes. Upon entering the S phase, the replication complex transitions into the replisome, coordinating DNA replication. DNA polymerase, epsilon, synthesizes the leading strand continuously in the same direction as the unwinding. DNA polymerase alpha initiates Okazaki fragments on the lagging strand by synthesizing 20 to 30 nucleotides. These fragments are further extended by DNA polymerase delta, synthesizing DNA in a discontinuous manner. pCNA, proliferating cell nuclear antigen, acts as a sliding clamp, holding the DNA polymerases in place during synthesis. RNase H removes most of the RNA primer, but one ribonucleotide remains attached at the 3' end. FLAP endonuclease 1, FEN1, removes this remaining ribonucleotide. DNA polymerase delta fills in the gaps between Okazaki fragments after primer removal. DNA ligase seals the nicks between fragments, creating a continuous lagging strand. Third, termination of DNA replication in eukaryotes. Termination of replication occurs when 2 replication forks converge. The exact location of fork convergence is not predetermined, as it depends on the timing and speed of fork progression. When forks meet, torsional strain builds up behind them and is resolved by topoisomerase II. The CMG helicase is thought to slide onto the last Okazaki fragment's double-stranded DNA during termination. A key step in termination is the removal of CMG helicase from chromatin. This is achieved through ubiquitination of the MCM7 subunit, followed by disassembly of the replisome by CDC48. Because eukaryotic chromosomes are linear, replication cannot reach the very ends of chromosomes. This is due to the need for an RNA primer on the lagging strand, which causes the loss of a DNA segment with every cycle. To prevent gene loss, telomeres, repetitive non-coding sequences, are located at the chromosome ends. In humans, this telomeric sequence is TTAGGG, repeated 100 to 1,000 times. Telomeres protect coding DNA, and only these non-essential sequences are lost during replication. Eventually, telomere shortening limits the number of times a cell can divide, a threshold known as the Hayflick limit. In germ cells, certain stem cells, and some white blood cells, telomerase enzyme extends the telomeres to prevent DNA degradation. Somatic cells, however, do not express telomerase, so their telomeres shorten with each replication cycle. Uncontrolled activation of telomerase in somatic cells can lead to cancer, allowing these cells to divide indefinitely. Proofreading. 3 replicative DNA polymerases, Pol alpha, Pol delta, and Pol epsilon, are responsible for replicating the eukaryotic genome. Pol alpha lacks internal proofreading activity, resulting in low fidelity during DNA synthesis. With an estimated base substitution error rate of 10 to the power of -4, Pol alpha contributes to the replication of approximately 1.5% of the eukaryotic genome, introducing hundreds of mismatches in each replication cycle. Pol D has exonucleolytic proofreading ability, which allows it to correct errors made by Pol A. Errors introduced by Pol delta and Pol alpha cannot be corrected by Pol epsilon. However, Pol delta can also proofread and correct errors made by Pol epsilon on the leading strand during replication. Evidence for semi-conservative replication of DNA in eukaryotes. In 1957, J. H. Taylor and P. Woods provided evidence for the semi-conservative replication of DNA in eukaryotes. They conducted their experiment using root tip cells of the broad bean plant, Vicia faba. The technique involved splitting root tip cells and using autoradiography along with light microscopy. To label the DNA, the cells were treated with radioactive thymidine, 3H-thymidine, which incorporated into newly synthesized DNA strands. After labeling, the cells were transferred to a normal, unlabeled growth medium containing colchicine, which inhibits sister chromatid separation during anaphase. The use of colchicine ensured that both sister chromatids remained together, allowing for easier observation of DNA distribution. In the first generation, both DNA strands showed even distribution of radioactivity, with one radioactively labeled strand and one unlabeled strand per DNA molecule. In the second generation, only one chromatid of each chromosome displayed radioactivity. This pattern confirmed that only one strand of the DNA in each new molecule came from the original labeled DNA, while the other was newly synthesized. These results supported the semi-conservative model of DNA replication, showing that each daughter DNA molecule inherits one old, parental strand and one newly synthesized strand. Difference between prokaryotic and eukaryotic replication. Origin of replication. Prokaryotes, single origin per DNA molecule. Eukaryotes, multiple origins per chromosome. Location. Prokaryotes, replication occurs in the cytoplasm. Eukaryotes, replication occurs in the nucleus. Rate of replication. Prokaryotes, faster, approximately 1,000 nucleotides per second. Eukaryotes, slower, approximately 50 to 100 nucleotides per second. DNA polymerase types. Prokaryotes, 5 types. Eukaryotes, 14 types. Telomerase. Prokaryotes, not present. Eukaryotes, present to maintain telomere length. RNA primer removal. Prokaryotes, done by DNA polymerase 1. Eukaryotes, done by RNase. H. Strand elongation. Prokaryotes, by DNA polymerase III. Eukaryotes, by pol alpha, pol delta, and pol epsilon. Sliding clamp. Prokaryotes, sliding clamp. Eukaryotes, pCNA, proliferating cell, nuclear antigen. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!